It's great to be here and to explain what is going on with regard to imaging and the ADRC at the UW. Here's what I would like to cover today. Talk a little bit about imaging biomarkers of Alzheimer's disease, which includes mainly the ATN framework that I'll talk about. How we're carrying on this imaging in the context of our research themes here. Talk specifically about the imaging and biomarker core and the specific resources that are available. And then my colleague Swati will talk about some of the specific studies that are happening here. So just to remind everybody as a touch point, of course, we know the hallmarks of Alzheimer's disease are amyloid plaques composed primarily of amyloid beta species and tau neurofibrillary tangles, which are intracellular accumulations of hyperphosphorylated tau. And you can think of plaques and tangles as having three levels of significance. First, they're the hallmarks of the disease. Just by definition, Alzheimer's disease is that which is accompanied by plaques and tangles and neurodegeneration. To the extent that we can detect and quantify amyloid and tau proteins in vivo, these can be used as biomarkers of disease. And at a third level, you think about amyloid and tau in their role in the etiology of the disease. What we want to talk about today is primarily the second level of significance, amyloid and tau as biomarkers of disease. And hopefully everybody is by now familiar with the NIA-AA framework for research diagnosis of Alzheimer's disease that leans on three axes of biomarkers, A, T, and N, A for amyloid, T for tau, and N for neurodegeneration. And this has been rolled out, I guess, over a series of meetings and consensus building meetings, and then increasingly sort of structured arrangements in the Alzheimer's Centers program. The amyloid markers are both imaging markers and fluid biomarkers. They don't exactly measure the same thing, and we can talk about that if you like, but there are ways to get at an amyloid marker with spinal fluid, or now blood plasma, or with specific molecular imaging with PET. Same thing for tau. Spinal fluid would be the gold standard. Plasma tau is coming on quickly, and tau PET scanning is got a great future, and we'll talk about that. And then neurodegeneration, which you can get at primarily using MRI markers of atrophy or hippocampal degeneration, but also has been thought of in terms of metabolic markers like FDG PET, or fluid markers like, I don't have neurofilament light there, but that's figures prominently in thinking about fluid markers of degeneration. And the ATN biomarker framework, as Cliff Jack and colleagues have described it, basically you have three binary markers, A, T, and N. If you have a positive A marker, positive amyloid marker, that puts you on the continuum of Alzheimer's disease. And if you also have a positive tau marker, T marker, then that's Alzheimer's disease full stop. And I've not made any reference to cognition, if you'll notice. So in the NIA-AA framework, you can define the presence of Alzheimer's disease without, with only a biologic measure. And then if you're amyloid negative, then you're in a regime where there's, and especially if there's N positive, you're in the regime of a non-Alzheimer's degenerative condition. That'll make sense. And the ATN biomarker characterization is increasingly being woven into the fabric of the Alzheimer's Centers program. And we'll talk about how that's working its way out here at UW. So the respective roles of MRI and PET imaging then, MRI is mostly used for structural imaging, diagnostic biomarkers, especially the presence and pattern of neurodegeneration, which can also be used as progression markers of disease. PET has two sort of modalities here. There's metabolic or functional PET, which estimates regional glucose metabolism, and that's a sensitive indicator of degeneration, and has been used as a diagnostic marker, especially when you have variant presentations of disease. And then there are pretty molecularly specific ligands that bind amyloid species or paired helical filament tau, and are useful more specifically for diagnosing Alzheimer's disease and its distribution in the brain. So in terms of use of MRI, one of the classic things we look for is hippocampal atrophy, since everyone's probably aware that Alzheimer's disease neurofibrillary pathology sort of initiates in the medial temporal lobe and gradually expands to other limbic areas, and then neocortical areas, especially association areas. So hippocampal degeneration is pretty common. I won't say universal, but virtually universal in Alzheimer's disease. And you can see on the left here the contrast of hippocampal appearance, sort of filling the temporal horn of the lateral ventricle in a cognitively normal person, giving way and accompanied by an enlargement of the temporal horn and mild cognitive impairment, while at a stage while most of the cortex is showing little atrophy. And then in an Alzheimer's dementia case, you see widespread atrophy, including profound hippocampal atrophy. And we can segment these structures in the brain using MRI algorithms, and even clinically, we can use computational tools that estimate the degree of that sort of volume of the hippocampus in comparison to the expectation for someone's age and sex, and same thing for the lateral inferior ventricle, the sort of temporal horn of the ventricle, and can calculate hippocampal occupancy as a sensitive measure of hippocampal degeneration. Cortical atrophy, likewise, is a measure of neurodegeneration, and we talk about the so-called MRI signature of Alzheimer's disease atrophy, which is a collection of brain regions which are particularly vulnerable to Alzheimer's disease, and can, even the atrophy in these regions can even predict decline in advance of any impairment. So those are just a quick illustration of how structural MR kind of factors into what we do. In general, those degenerative changes that we see with MR are late phenomena. This is the classic, now classic figure that comes out of the ATN work from the JACC lab, showing what I call a canonical sequence of Alzheimer's disease biomarkers over the disease course from the latent pre-symptomatic stage through amnestic mild cognitive impairment and dementia. The markers which take off first are measures of amyloid accumulation, and tau is close on the heels of amyloid changes. MRI and FDG PET, which indicate degeneration, are showing up somewhat later, and cognitive impairment still later. So there's a big latent and pre-symptomatic phase, probably 15 years or so, during which these markers are positive, but there is no apparent impact on cognition yet. One of the big interests in the biomarkers, the ATN biomarkers, is this is a way of approaching pre-symptomatic disease. right? You want to find a way to define disease that doesn't depend on cognitive change if you want to detect it and study it in this window of opportunity before there is dementia. And then we talk a lot as a center, as you've probably noticed, about the phenomenon of resilience or cognitive resilience to Alzheimer's disease. And this means that there are some brains tolerate a burden of Alzheimer's pathology better than others, function better than others in spite of pathology. And the way one can get at this depends on an imaging approach. The so-called residual method, in which you look at the relationship between a measure of cognitive function and a measure of brain health, for example, total brain atrophy. And there's a regression line that fits there, and the residual is above or below the line by definition. And for individuals with a residual below the line, they are thought of as having low degree of resilience, or the flip side, vulnerability. And for those who are falling above the regression line, that residual is an estimate of their degree of resilience, if you will. And that residual fits, will show a relationship to things like level of education. These are data below that come from our Memory and Brain Wellness Center, or you could think of other things which are related to, you've heard about related to resilience, besides educational factors. That's the way to approach resilience sort of from a biological perspective. Structural MRI itself is not a sensitive and specific modality for detecting Alzheimer's disease degeneration. I've sort of already said that, but the reasons are that there's a long preclinical latent period that you can't radiologically distinguish from normal aging. This varying resilience, which you have a situation where you have different degrees of degeneration that happen before people develop symptoms. And there's variation in the pattern of neurodegeneration across individual cases of Alzheimer's disease that causes it to overlap with other diseases. So MRI alone is sort of not sufficient for tracking Alzheimer's disease. So talk a little bit about the PET modalities then. So FDG PET. So what I'm showing you here is a FLAIR MRI sequence, and this is someone with memory loss. And most radiologists will look at this and call this normal. I submit to you this isn't normal, but it's only with a practiced clinical eye that you can detect that there's some relative atrophy in the left parietal lobe relative to the right parietal lobe. But if you do FDG PET, you can see a clear-cut reduction in tracer uptake in that region. And then we further process the data, at least clinically, to generate these statistical parametric maps that show regions of low metabolism relative to the expectation of a normal group. So here is an example where the PET scan makes a diagnosis of Alzheimer profile neurodegeneration pretty clear cut, whereas the MRI scan was subtly abnormal at best. So that's why we like FDG PET. For the molecular PET, we have two species of ligands that we're using. So there are amyloid avid ligands like Pittsburgh B or florbetapir. And here are some examples of amyloid PET scans. So in the green boxes are normal scans, and in the red boxes are abnormal scans. And to make sense of this, what you need to know is that, MRI is on the bottom, of course, and PET scans on the top. But what you need to know is that there's a non-specific uptake of tracer into white matter. And so gray is tracer. You can see that in a normal scan, you can sort of see the white matter skeleton there. You can see the white matter core and the callosal fibers and so forth. And then you have interspersed fingers where the gray matter are, the gray matter regions are. If you look at an abnormal scan here on the right, you can see that sort of gray-white distinction is obliterated, right? And you see this sort of ground glass appearance where you can't really see the gray-white contrast. That's what defines a positive amyloid PET scan. clinically. And you can also measure this quantitatively, but a visual read is sort of, at least in the clinical realm, all you need, because by the time someone comes in with amnestic mild cognitive impairment, it looks like these scans on the right. It's not a close call. Tau ligands show a more variable uptake, but tend to correlate pretty closely to the clinical variability that we observe. But show uptake in medial temporal regions and association area regions, as sort of in harmony with what we know about the distribution of neurofibrillary tangles. So you have a normal scan above with someone using the MK6240 tracer, which we have been working for years to get to stand up at the UW, and someone below with an abnormal scan. And if I were to summarize, the impact of these molecular imaging modalities, I would say that amyloid PET scans are principally useful to certify diagnosis and identify preclinical Alzheimer's disease. The uptake is pretty stereotypical. It varies there's no appreciable variability from case to case in terms of a hippocampal sparing case or a hippocampal predominant case, for example. You could use a fluid measure just as effectively as an amyloid PET scan in general. Tau is another story. There's a variable distribution that correlates well with symptoms and does track advancing disease pretty well. So I see the future of tau PET scan as a progression marker. We can talk about that if you like. There have been some really important large-scale imaging studies in the field that we deal with on a regular basis, so you should be familiar with those. The Alzheimer's Disease Neuroimaging Initiative was kicked off in 2004 and is now continuing into its fourth phase, so ADNI-4. This is pretty much set the standard for defining the ATN biomarkers. It's pretty much set the standard for open neuroscience as well. The data from the ADNI study were available freely after quality control and before the investigators had any special access to them. This open science standard was later picked up by the Allen Brain Institute and its work, and many other studies that you're familiar with now have followed suit. So it's an important thing in the imaging field that there are these large public data sets that are available to you as an investigator, as much as any other investigator in the world. A lot of questions have been asked or generated using ADNI data. So that's something you should know about. The SCAN initiative, SCAN stands for Standardized Centralized Alzheimer's Neuroimaging, related to ADNI in that its SCAN is using the same imaging sequences that ADNI has developed, but is more specific to the Alzheimer's Centers program, and it's a scaled up version of ADNI. So it's a U24 mechanism project that is enabling neuroimaging across all the Alzheimer's centers, including ours. These data are just starting to become available at scale for people in the centers program, but the first data dump through the Laboratory of Neuroimaging in California and the National Alzheimer's Coordinating Center is going to be available to us this summer, to everybody on this call this summer. That includes something like 500 tau scans and 500 amyloid scans, and I don't know how many MRI scans. So it's fairly sizable just in a first installment. But if you think about there being thousands of participants in the NAC funded or the NAC conforming studies across all the Alzheimer's centers. And when you think about the Clarity grant that I'm going to tell you about in a little bit, this number is going to rapidly go up into the many thousands. So this is going to be a big resource. So that's what I want to say in terms of biomarkers. In terms of our center, as everybody's aware, we're focused on mechanistic and biologic underpinnings of Alzheimer's disease and also the factors that counter degeneration dementia. We're also, as a center, very interested in novel biomarkers and early diagnosis, which takes us to imaging. Increasingly, groups like Jeff's and Swati's are studying vascular factors in Alzheimer's disease and related dementia. So that's emerging as a theme. Frontotemporal degeneration, and then we have a nationally distinctive program looking at ADRD in American Indians. And our resources, you've probably seen our so-called square diagram, which describes how we organize the resources in the center. There's a kind of large potential population that we recruit from. So the Memory and Brain Wellness Center at Harborview, the ACT study, a potential participant registry that we maintain, and the focus there is on early detection and identifying people that we would like to prioritize for bringing into a deeper and standardized characterization in the clinical core. And this is really the centerpiece of the clinical research component of an ADRC, the so-called clinical core, and its longitudinal cohort, which numbers 400 or 500 individuals and gets characterized with to national standards. So so-called uniform data set, ATN, biomarker characterization, and standard approaches to brain autopsy eventually. And in our center, we think about this in terms of looking at the clinical variability of the disease and how these biomarkers distribute over space and sort of correlate with cognitive and other variability in Alzheimer's disease. And then subsets of the participants in the longitudinal cohort then are studied more intensively with other methods. For example, we're doing precision autopsies that are aligned with work we're doing with the Allen Brain Institute. Jessica Young and her colleagues are looking at iPSC and organoids from selected participants. My group is interested in the topographic variability of the disease and so forth. And then sort of inserted in there, we have the Native Research and Resource Core and the prior satellite core, the previous version of the Alzheimer's Center that's got a lot of data concerning American Indians and Alaskan Natives, and that's a significant resource as well. I want to talk about the variability of Alzheimer's disease. Just going to give you one example. So this is typical Alzheimer's degeneration, sort of hippocampal atrophy, cortical atrophy, especially posteriorly. And you can see this kind of summarized in this 3D diagram. And yet there are cases which show variation on this central tendency. So here's an example of someone who has significant medial temporal atrophy, but very little, if any, parietal atrophy. And here's another case of someone who's got very little medial temporal atrophy, but a lot of parietal atrophy. And these are so-called limbic predominant and hippocampal sparing subtypes of Alzheimer's disease, and they're each about 15% of cases. And they this kind of variability is an important clue to variation in the underlying biology of the disease, and that's one of the things that we're interested in. The imaging and biomarker core of the center has got a lot of participants on the investigator team, and you hopefully have met Dr. McDonald, who's the co-leader and is really the mastermind of the postmortem imaging part of the center. And Jeff, has Christine done, and she hasn't done an AD at UW with us
Yeah, she frequently ends up with the neuropath group. So she was
Okay.
part of the neuropath presentation a while back.
Okay. All right. So people have access to that information. And then Swati will be our next speaker, and is a bioengineer with expertise in full sequence programming and image processing, and a lot of cross-disciplinary expertise in ADRD, which is a great combination. So you'll hear a little bit about that. So I don't want to dwell on the aims, just to say we're trafficking in MRI and PET data and ex vivo MRI data. We're ideally assigning an ATN classification to all of participants in the center. That the way that we do that has evolved significantly over the last few years as plasma markers have become available. And as we've struggled with the impact of the COVID pandemic, which made it hard to bring people in and get spinal taps and things like that. And then we're providing expertise in all these areas to people that are doing science that needs imaging at the UW. So there's a number of innovative approaches as well that we make available. Swati may talk about some of these. We can measure cerebrovascular reactivity, which is a way of looking at the integrity of the physiology of the small vessels. We can image myelin water by a couple different approaches. We can now efficiently generate co-registered images of the underlying physical parameters of MRI, T1, T2, and PD in an absolute sense, and synthesize any MRI contrast you might want for your study. That has interesting applications and is, and I'll mention it a little more detail. And a lot of expertise in image processing that I may not have time to get to. That quantitative or synthetic MRI approach that I mentioned a second ago works something like this. There's essentially a curve fitting exercise that happens as an integral part of the pulse sequence that generates images, sort of fits and estimates and then generates images of the underlying physical parameters that determine what an MRI image looks like. So you've heard of T1 weighted images that are weighted according to the so-called T1 relaxation time, but these are different. These are images of actual absolute T1, which is independent of, it's a biological property. It's independent of the pulse sequence or the scanner that you do it on. And then you can mix T1, T2, and proton density to generate the standard contrast that you typically see in a clinical MRI study. And we're doing a project called the Deep Neuro Project, which is looking at using these sorts of quantitative images as a way to more easily harmonize data across different centers and different scanners in large-scale studies. One virtue of this synthetic MRI approach is that it only takes 6 minutes to get all that data. So it's very efficient and potentially helpful in scaling studies where you're interested in MRI as a primary measure. And that's when the software processes this synthetic MRI data, you get something like this, and you can actually put, someone's data point on a normative curve that looks something like a head circumference curve, but this particular participant who's 60 years old has a gray matter volume, which is squarely inside the range of normal for that, for the population. You can do the same thing for the white matter fraction, and it's pretty great. If you've paid attention to the neuropathology piece, you realize that we're extensively sampling the brain, more extensively sampling the brain at post than other centers have typically done. And this is really important in some of the emerging work that's being done with the Allen Institute, for example. And we've strived as a center to match that kind of anatomical precision with the way we sample the imaging data so that we can generate rectangular data sets that give you imaging measures in vivo of the same regions. We're going to then talk about ex vivo, and there's a lot of effort in the background gone into like co-registering those images and the tissue so that you can be confident that you're sampling the brain areas that you think you are. And so this is just summarizing that we have in vivo imaging measures that I've been talking about so far, but they have their counterparts in the ex vivo program that Christine is leading. So thinking now more specifically about what we have, the Memory and Brain Wellness Center clinic has had 10,000 patients or so over the years. There is an IRB approved repository of those data, and we have curated at least 700 data sets that have a research grade MRI scan along with solid diagnosis, cognitive data, and other neurologic data. So that's a great data set that you can use for generating hypotheses. It's not been collected for research purposes, but it's available. Then we have about 1,000 individuals that are extracted primarily from the 10,000 that have indicated in principle interest in research and are part of a research registry, many of whom have been recruited into the clinical core. In our center, we're seeing about 30% of our clientele is having mild cognitive impairment, which is right now the target population for enrollment in the clinical core. And then in the clinical core, as I mentioned earlier, we are aiming for 500 participants. We currently have around 400 participants that are sort of standardly characterized with the uniform data set, ApoE genotyping, scan compliant MRI, ATN, and the ATN piece is, I think the next slide might talk more about that. Let's see. Yeah, here. So how are we assigning ATN in the center using imaging and fluid markers? So we describe the process by which we analyze the available data for each participant, which might come from lumbar puncture or blood or imaging as an ATN exercise in ATN consensus. And we have a committee that draws from several disciplines to do this and articulate what is the algorithm for it. In our sort of pilot study with this, we worked with 115 individuals that were studied in the last cycle of the center with fMRI and spinal fluid biomarkers, and we have evaluated using the biomarkers to triage individuals to those who are amyloid positive and are therefore people we want to look at with tau PET to characterize their sort of anatomical profile degeneration, those who are amyloid negative, for whom it would not be a good idea to do a tau scan since it's going to be negative and it's just a waste of money, and whom we follow with MRI and plasma markers over time. And then a borderline group that either has borderline amyloid values or conflicting information like amyloid negative but tau positive, that we would then do a follow-up amyloid measure with amyloid PET to determine are they in the amyloid positive group or not after all. And the numbers work out that about 30% of people would go on to having a tau PET scan. And about 20% of the cases have reason to come to are not confidently slotted into one of these categories without the need to appeal to the committee. So about 20% of the cases need to come before the committee. And what we're going to do going forward is, instead of leaning on LPs alone, we're going to screen first with plasma phospho-tau, and plasma p-tau acts like a great surrogate for amyloid positivity, actually, and there are pretty good data for that. So those who are p-tau positive will go on to have a confirmation with an LP or an amyloid PET scan, and those are negative will go to the MRI monitoring file. And those who are confirmed to be amyloid positive will go on to tau PET. So that's our strategy moving forward. And I'm almost done. I have a couple slides. Just want to tell you about some recent developments. How am I doing on time? Pretty good. So the first thing is the Clarity Consortium. So Clarity is an acronym that stands for the Consortium for Clarity in Alzheimer's Disease Research Through Imaging. This is a massive R01. I mean, this is a $100 million R01 that was written by colleagues at Wisconsin and Stanford on behalf of the Alzheimer's. These are the leaders of the current sort of steering committee for imaging for NAC. And on behalf of NAC and the Centers Program, they've written this R01 that will fund ATN imaging for our clinical core and the clinical cores of all the other P30 centers in the network, to the tune of a total of 2,000 participants recruited over the next few years, which will be imaged twice. The idea is to recruit 60% impaired and 40% unimpaired. We are expecting that about 75 participants in our center will have their imaging funded by Clarity. And these data will conform to the scan standards and will be distributed via NAC and the mechanisms that I mentioned a little while ago. Besides being a center that's going to benefit from Clarity, we also have some specific roles in Clarity for our center. So Dirk and the Neuropath Core are serving as the Neuropath Core for Clarity. So people that participate in the Clarity study that come to autopsy the tissue will be sent back to Dirk's lab for analysis. Swati has a leadership role in the advanced imaging sequences component of the grant. She may describe that a little bit more, but as a typo here, but there's going to be some, centers can opt into using some innovative techniques, including motion navigated T1 scans and arterial spin labeling scans. And one of our junior faculty, Dr. Jahanian, also has a role on the project. And Swati is especially important to bringing all the Philips scan sites kind of technically into line with the program. And then I'm also co-investigator on the imaging data analysis component of Clarity. So Clarity is a big win for us and for the whole program, and it was just announced that it's been funded. So we've got the just-in-time request day before yesterday. And then I want to just say a word that we're on the cusp of potentially a different phase of Alzheimer's disease work, clinically speaking, because the FDA is poised to approve lecanemab, which is an amyloid removing antibody. I think the VA has already approved lecanemab internally. I don't know if our VA is actually doing any work in that direction yet, but we're expecting over the next year, we're going to start to roll this treatment out to pretty highly selected subset of cases, but they happen to be the same cases that we want to recruit into the ADRC already. So people with amnestic MCI that's got a positive amyloid biomarker. And we're going to, this is, opportunity and also a lot to think about. So we're still really thinking about how this is going to work. But one of the things that happens when we treat patients with high dose lecanemab is it does actually reduce the amyloid burden. You can see that this is a dose-dependent reduction in amyloid ligand, and your amyloid scan does literally become negative over a 12 or 18-month period, and then comes at the risk of consequences of removing amyloid from the small vessel walls, which include extravasation of fluid, so-called, there's this acronym ARIA, amyloid-related imaging abnormalities, but ARIA-E is the sort of edema version, and then microhemorrhages around these small vessels. The ARIA-H version are well-recognized risks of this high-dose antibody therapy. And most of the time, this is asymptomatic, but a small portion of the time, this becomes a clinical problem. So these kinds of participants are almost surely going to be coming into the clinical core in some number in the coming few years. So that's hasn't started yet, but it's also an opportunity to do something systematic with this group, either here or across the center's program. So ways to use these resources. Of course, you can tap expertise in the core to help design your study if you need an imaging piece. You can access the imaging data in the Memory and Brain Wellness repository that I described. You can access our observational cohort imaging data as well. You can do this directly from us or indirectly through NAC. And then there are public and in silico discovery databases out there. I mentioned ADNI. I mentioned the SCAN database, which is coming online this summer. We are going to be providing ATN characterized participants, so these are potentially recruitable for your study. You don't have to repeat that characterization. And if you have a larger study, this can integrate with, even drive the composition of the clinical core to some extent. And you can apply for an ADRC development project that takes advantage of these resources.